So today we have our Unimind meeting where we discuss the interconnection between the gut-brain axis, the microbiome, and psychedelic experiences. And yeah, as you all may know, we are joined by Pam Gonzales. Pam holds a PhD from the University of Groningen, is also the co-founder of the Psychedelic Society Groningen, and also involved in Unimind Groningen. So yeah, in the context of her PhD, she investigated the molecular interactions between the gut microbiome and the host through neuroactive compounds. And today she will merge her interest in the field of psychedelics with her passion for the microbiome to give us some insights into the interconnections and relationship between the gut microbiota, gut-brain axis, and psychedelic experiences. So yeah, with those words, I would like to give it over to you, Pam.
Thank you so much. Thank you so much. Thank you so much. First of all, I'm very, very happy and excited to be here to giving this talk, merging these two beautiful worlds, which I really feel very passionate about, which is about, I'm gonna share my screen. Which is the microbiota, the gut-brain axis, and of course, psychedelics. We will today explore the interconnections between the gut microbiota and psychedelic experience. As mentioned before by Stephen and Pamela González, that is my email if you would like to contact me later on. And I'm very, very thankful with all the organizers, Anna, I always forget his name, and Stephen, for the opportunity for being here today, Unimind Maastricht and Groningen, and the Psychedelic Society, that we joined forces together to bring this talk here. And because it's related to this talk, I normally don't talk too much about myself, but I would like to give just like my scientific background. I am Mexican and I did my bachelor studies in Mexico City. My bachelor in science is on biotechnology, and I have a specialization on molecular diagnosis. And that's when I start to realize that I really love the molecular part of things. And actually, when I was in my bachelor's, I tried for the first time, I had my first psychedelic experience. And then I thought like, I needed to know more about this world. So how can I help and how can I understand it better? So that's why I decided to come to the Netherlands and study my master's on molecular biology and biotechnology. I joined the top program of biomolecular sciences, and my two master's internships were about the gut microbiota, one in the University of Groningen, which basically I was looking for neuroactive compounds that the microbiota can degrade those compounds, and I was looking for proteases. And my second project was about Faecalibacterium prosnitzi, which is one of the butyrate producers bacteria and a specific gene. I was doing research in France, in Rye. And after that, I really like all of that. And I decided to come back to Groningen to do my PhD, also in molecular biology, biomolecular sciences, in the host microbe interactions department, in which I got graduated actually 1 year ago. And part of my studies, my in vivo studies in mice, I did it in University of California in San Diego. And there I was doing research about a gut peptide that actually, alters or let's say it regulates the composition of the gut microbiota and how that was affecting the gut-brain axis. And in the last part of my PhD, I was so lucky to be the chair of the first ever psychedelic summer school in Groningen. So I was super proud to be planting that seed of this beautiful project that next month it will be also taken, it will be happening in Groningen. So I know that there's also people here from the new organization. I'm really proud of you guys that now you are organizing this really beautiful event. And last year, meeting people in the Psychedelic Summer School led me to know other people, which we together gather, and we thought like, yeah, there must be some kind of psychedelic society. There's already people talking about psychedelics. And we together co-founded the Psychedelic Society of Groningen. But of course, as a psychedelic society, we are all part of it. So, and that's why I'm also here speaking also on their behalf. And very quick, what we do is basically, we are an organization, and we would like just to revolutionize that stigmatized idea on psychedelics and just put our pinch of grain on the re-emergence on psychedelics in a well-founded manner. So for that reason, we disseminate science also. So that's why we're here with Unimind. We also do integration sessions just to reflect upon our important experiences of our lives, not only about psychedelics, but we also have other activities such as mindfulness, like cacao circles. We also involve yoga, breathwork, or meditation. And we are constantly giving sessions in which we talk about psychedelics to reduce harm and risk, just to navigate safely during our psychedelic experience. And if you would like to follow us, there are some channels. So thank you so much for that. Today, I will talk about the gut microbiota, which are microbial factories. Then I will talk about the microbiota gut-brain axis. Then we will talk about psychedelics and the gut microbiota, some perspectives about it, and some conclusions, the scientific ones, but also philosophical conclusions. And here I would like to include all of you, like, this floor is open to everyone. And actually, this talk is being recorded, and as Steven said, it will be only recorded until I finish my talk, but the discussion won't be recorded. And yeah, of course, if there's any questions, any remarks, let me know. And if there's something that you don't understand during the presentation, if it's a quick question, feel free to put it in the chat. So let's start. So first of all, here you can see my pointer. What's the gut microbiota? So it is also known as the microbiome, which is the vast diversity of microbes. They're not only bacteria, but it also comprises virus, fungi, archaea, protozoa. And as you can see here, we have trillions of them. Actually, like the number of these bacteria, it's higher than the number of cells that we have in our body. And the microbiome is the collection of all these genes of the microbiota. And as you can see here, these genes surpass the genes of the human. So they have a great capacity of metabolism. And we have a symbiotic relationship with the gut microbes. And that means that as we let them live in our cells, they also benefit from us by having nutrients. But of course, we really need them, because without microbes, we wouldn't be here today, right? Because great part of metabolism, our immune function, our neurocognitive functions are because of microbes. Each person has their own gut microbe. It's like our own fingerprint. And there's many factors that I'm going to talk about later on how this microbiota is very, very diverse. And this field is, well, some people say it's new, but it's already more than 20 years that the field is here. And I only know a little bit because it's also really, really broad. You will see why later on. And yeah, microbes are really essential to our lives. We couldn't live without them. Like they regulate ourselves. And here in this picture, you can see that how why our gut microbiota is really different from each person. First of all, we will have differences depending on our diet. If we will have fiber-rich diets, or we have high-fat diets, that will also be a difference on ourselves. If we take any kind of pharmaceuticals, supplements, probiotics, or also any other kind of pharmaceuticals, it also depends on our origin, our ethnicity for our whole genome. It also depends in which stage of our lives we are. So most of the time, like in babies, the microbiome starts to develop when they are born. And it also depends if you are born by a C-section or vaginal delivery. That's when everything starts. It also depends if your mom gave you breastfeed, you were breastfed, or you did or you were not. So all of that, it alters our composition of our microbiota. And also if you've taken antibiotics or other medication over your growth, that will also impact on your microbiota footprint. And of course, and very important, stress, how we manage stress, if we are active, how is our pattern of sleep, that also influences a lot our gut microbiota. And it is important to have this into consideration for later stages of the talk. And as I said before, the microbes are microbial factories. That means that they produce molecules that will benefit our cells, but also if there's no balance, that can lead to disease. So in this picture, you can see how the gut microbiota is involved. For today, we're gonna mostly focus on the gut-brain axis, the psychiatric disorders, and how an unbalanced gut microbiota can lead to disease. As you can see, there's other like axes that in the end, I think that all of them are interconnected. And all of these illnesses can be also dysfunctions. Sometimes when you get to an imbalance, you can get back on track to homeostasis, to balance if you change your lifestyle or your patterns of behavior or yeah, how you live, basically. And we also have comorbidities, and comorbidities are the combination of several diseases together. So, for instance, some people that have metabolic diseases like diabetes can have also some mental disorders, or people with depression, sometimes they can have metabolic disorders or obesity. And this is because we will have certain inflammatory state. So for this reason, the field is very, very broad. We're all the time looking for novel and effective therapies. And in this case, we have two very beautiful options, either the microbial therapies, which most of the time involves the use of antibiotics, but also fecal microbial transplantations or probiotics. These are like microbial therapies, and psychedelic therapies, which is basically the use of psychedelic substances to treat any disease. And in this case, now science is mostly approached to mental diseases because of all the regulatory situation on psychedelics right now, because there's increasing the scientific outcome on psychedelics. Hopefully we were gonna get also to change the legal situation, and that will build into more research. So first of all, to understand all of this, I would like to give a brief introduction on how our gut is, how it is made. So we have layers in our gut, and we mostly have 4 layers, which is the serosa from outside to inside, is the serosa, the muscularis, the submucosa, and the mucosa. And basically, like, the serosa is the layer of the gut that contains all and gives shape to our gut. All of them give structure, but all of these layers are made by very specialized tissue. As you can see here in the muscularis, we have muscle, and here they're also in between the enteric nervous system, which are nerves around all this muscle. And here we also find neurotransmitters that involve this gut motility. However, the motility doesn't start just here, but it actually starts from the gut lumen. And actually, it comes from signals that the gut microbiota provide to our mammalian cells. So there is interkingdom communication. Then afterwards, we have the submucosa, which basically is where all the immune cells are. And after that, there's also the mucosa, which we have immune cells and also epithelium. Here's another picture in another angle. As you can see, here is the serosa, then the muscularis externa, the submucosa, and the mucosa. And here on top, so here in the middle, there is where all the gut microbiota is living. So basically it's our lumen or the gut content. And there's also the feces if it's in the colonic or part of our gut. And these little villi and glands are in contact directly with our gut microbiota. And if we zoom in, let's explore the molecular interactions. Yes, so here I would like to start talk about, if you can see first here, we have the gut wall, which is basically that physical barrier that prevents the bacteria to translocate to the inner part of our body. Here, we see that the gut microbiota produces several kinds of metabolites or molecules that will actually affect our brain. And this is how we call the microbiota-gut-brain axis. For example, in this gut wall, we have specialized cells. It's a really big tissue. Actually, I think it's the largest tissue of the human. And we have cells of different kind. For example, we have enteroendocrine cells, and these cells produce hormones that basically will regulate our mood and also our satiety. That goes directly to our brain and our behavior. Moreover, we have here immune cells, and immune cells produce cytokine or other inflammatory molecules or anti-inflammatory molecules that will have effects on our gut epithelium, but also in the enteric nerves. Moreover, we also have the enterochromaffin cells, and those cells are the ones that that produce serotonin. And most of you should know already by now that most of our serotonin, 90% of our serotonin, is actually produced in our gut. And the microbial cells actually have a huge, huge role in this synthesis. And for that reason, also serotonin is involved in inflammatory states. And also it is involved in the gut motility. So here we have another overview. And it is important to note that these are multidirectional interactions. It's not that there's only one way or two ways. Actually, everything is happening at the same time. It is a very, very complex system. And it is still like ordered in a certain way. But of course, sometimes this can get affected due to our lifestyle, to disease, infections, or to stress. Here we can see that this little part, this is a zoom in of what I already showed you. This is the gut microbiota, this is the gut wall. And here we have a layer that is also very important, which is a layer of the mucus layer. And that is actually what it doesn't let the gut microbes to penetrate the inside of our cells. Because if there's any kind of LPS, so bacterial membrane that passes this barrier, immediately our immune system is going to send signals saying that there's something wrong, and of course you will have inflammation. And of course that will impact our other organs. And in this case, we also have here, for instance, the liver. In the liver, it's also very important. We have the bile acids, and the microbiota contributes during a secondary metabolism of bile acids, and basically it's where we detoxify everything. And here is also the kidney, and the kidney and the vagus nerve is really important for the regulation of stress, because when we are stressed, we actually feel, we release cortisol, and that for sure will impact in the levels or in the composition of the gut microbiota. And we have this fight or flight response, in which, yeah, sometimes when we feel fear or we are like alert on something, our voice is like getting a little bit like this, or our heart rate raises, and our gut feels also like butterflies. And all of this is because there's some cortisol going on there, and it's a signal of stress, and cortisol affects greatly the composition of our gut microbiota. And having an unbalanced gut microbiota, we already said before, that can lead to dysfunctions or in dramatic cases, illnesses. So these microbes regulate our physiology, because all of these interactions depend on each part of, let's say, the members of this system. If we wouldn't have gut microbiota, we wouldn't have a really good immune system. Thus, we wouldn't have the communication that are going on here with the enteric nervous system. Then we wouldn't have the communication going to the brain, to the liver, and also to the periphery. As I said, a great part of our metabolism is done here, and actually the gut microbiota is, I don't know why it's not considered another organ, but it's even highly important as an organ. And for that reason, we have neuroimmune responses and also psychological behavior, and mood is also highly involved in here. And for that reason, yeah, before passing here, for that reason, I would like to talk about microbes, right? Like, what is this microbial metabolism of these neuroactive compounds? As I said before, the enteric nervous system is also known as our second brain. And it is our second brain because actually a great big deal of neurons are there. Like some people say like it's 1/3, some people say like we have even more. So it is not clear yet, but it is true that we have a lot of neurons in our gut. And we also have the blood-brain barrier that actually controls the passage and exchange of molecules and nutrients between the blood circulation in the periphery and the brain cells. So the physiology of the blood-brain barrier is really important, and there's actually evidence in germ-free mice that the lack of microbiota creates permeability in the blood-brain barrier that can also lead to disease. So here, as you can see, in another perspective, we have the Microbiota-Gut-Brain axis. We have neurodevelopmental and neuropsychiatric diseases here, but also neurodegenerative diseases linked to any deficiency happening here. We have microbial metabolism, in which also receptors, the mammalian receptors are here involved, and neurotransmitters as well. We have several pathways involved, from the immune system, the endocrine system, neurological system, synthetic nervous, the circulation, the vagus nerve, and we have several molecules that regulate all these pathways. Regarding the microbial metabolism, we have like a huge variety of molecules, but I'm going to just mention the ones important for this topic, which are the short-chain fatty acids. We have many different short-chain fatty acids, but you will mostly hear about butyrate, propionate, and acetate. And actually, these three molecules give the shape, and they make clear and sure that our constitution of our gut layer is in place. And here you can see a picture of the leaky gut. I don't like this name, but it's really well known that when we have an inflammatory state, we have here proteins in between all the tight junctions, that when we don't have inflammation, we have everything in balance. But sometimes when there's translocation of bacteria, there are some proteases or enzymes that degrade these tight junctions. And this disbalance can also be due to the huge number of inflammatory molecules or a huge number of neurotransmitters that are not in balance. So all of this system is dysregulated, and it goes to our blood circulation that will, of course, impact our brain. And we also have the tryptophan metabolism. So this amino acid is really, really important because there we will have pathways related to kynurenine. I'm going to talk about this later on. Of course, about serotonin and melatonin, that is a hormone or neurotransmitter that regulates our sleep. Serotonin, it's really important for our mood, for our behavior, but it's also a very important neurotransmitter for our physiology. It regulates our gut motility. It also regulates relaxation of our muscles. So it's not only important for our mood and behavior, but also for our physiology. There's also indose and the tryptamine itself. So after that, how do you think the microbiota is connected with the psychedelic experience? So first of all, I would like also to introduce you to the microbial metabolism of drugs. And I'm going to give an example from the department that I used to do my PhD. There, one of my colleagues did research about the treatment for people with Parkinson's disease. And basically, Parkinson's disease is a neurodegenerative disease of dopaminergic neurons. Either they have any anomaly in their shape, or the neurons are dying. So there's a lack of dopamine in our body. And for that reason, all the processes regarding cognition and also physiological functions are disturbed. And the treatment for Parkinson's disease is levodopa, which is basically L-dopa with another enzyme that prevents the degradation of L-dopa. However, my colleagues found that there is a microbial conversion in the gut of L-dopa, and it actually happening in the small intestine. So once people with Parkinson's disease take the medicine, the L-dopa gets converted immediately into dopamine. And why are people with Parkinson's disease taking L-dopa and not dopamine? And that is because L-dopa can close the blood-brain barrier, but not dopamine. So dopamine stays in the gut, and that's why we have higher levels of L-dopa or dopamine, which can cause the side effects that people with Parkinson's have, which is mostly the dyskinesia, so the rigid movements. And actually the treatment is that they start taking certain dosage, but over the time they are increasing also this dosage. And it is just really surprising that pharmaceuticals are not taking into consideration the microbial metabolism of drugs, no? And it's really, really important because, as we said, like each person has their gut microbes very unique, and what can work for some people cannot work with others due to this really important reason. So, of course, the therapeutic property of the drug is diminished, and it's really a shame for the people that are taking it, right? So here is a scheme on how the drug works. So if we have members of the gut microbiota that have high levels of this enzyme that degrades that converts L-dopa into dopamine, then there's no dopa going into the blood and the bloodstream. Thus, we won't have the therapeutic effect. Then now, after all this kind of background, what is actually known in the field of psychedelics and the gut microbiota? So first of all, I think that mostly it is it's a field that is not fully discovered, and it's just really, really in early stages. And what I found mostly was regarding ketamine, and why ketamine, because also it's an anesthetic, that it's not really banned. Of course, for handling it's for medical use, but then there's people and research who do research with ketamine, and it's a typical psychedelic, so it's not among the classic psychedelics. And classic psychedelics are the ones that bind specifically to serotonin receptors or through serotonin pathways, and it is actually anesthetic in high doses, but recently there's a huge evidence pointing out that it also has antidepressant properties, and that can be also related to its anti-inflammatory properties. I forgot to mention, but also in our gut we have a huge glutamatergic system. And ketamine binds to N-methyl-D-aspartate receptors and MDA receptors, and it's a dissociative agent. So what I found is they did experiments, in vitro experiments, so just in petri dishes in which they tested ketamine against several strains, and they found out actually that ketamine is antimicrobial, so it means that either it doesn't let bacteria grow or it kills the bacteria. And most of those bacteria were gram-negatives, so especially pathogens have this gram-negativity, let's say. However, this is just not really translatable to the real situation because these high doses wouldn't be used in vivo. So this is just, it's just good to know. However, when we have dysbiosis, as I mentioned before, we have altered gut microbiota, and there are studies that also show that BDNF, which is like brain-derived neurotrophic factor, I'm going to talk about this factor afterwards. And the receptors in which ketamine bind are also imbalanced, or their activity is not the best. So in this review article from Wilkowska, there, I really like it. I would recommend that if you are interested in the topic to take a look on it. But they discuss basically how treatment with ketamine, especially in rodents, have altered the gut microbiota composition, but also the gut microbiota I found a thesis. It is not peer-reviewed, it's not published yet. I just found a thesis there from the University of Miami, in which this person did the studies with psilocybin, and actually in the same group, they have some recombinant strains that synthesize psilocybin, so from E. coli. And he used this psilocybin and a precursor of psilocybin to do his experiments, and in rats. And he also found out that in certain concentrations of psilocybin, the gut microbiota changes the composition. So for that reason, now I just think like if the anti-inflammatory reaction of psychedelics is also through the balance of microbes. This, of course, is not known yet. Yes, and yeah, I will continue. And of course, there's a big, let's say, capacity of, the ability of creating new neuronal connections or functions, or creating new neurons, new neuronal cells. It's called neurogenesis. And psychedelics and microbes are known for this capacity. So first of all, I would like to talk about BDNF. There's already literature pointing out that gut microbiota is linked to this BDNF. BDNF, it's supposed to be, or now there's a great deal of evidence that it can be a big player in neurogenesis, actually. And there is this paper, the first one, in which they found a direct link between certain members of the gut microbiota that when they are there, which are mostly butyrate-producing bacteria, they promote BDNF activation or its signaling, promoting neurogenesis. So it's really beautiful. And I found other two papers pointing out to the same thing with different members of the microbiota, also butyrate-producing bacteria. So it seems that one of these short-chain fatty acids, it's actually helping with this neurogenesis. And why butyrate is known to regulate great deal of our genes, because it is in epigenetics. We need a butyrate to unfold the genes, so then transcription can take place. And there is a recent study, which I was super happy to read, which is the last one, in which they also found that the classic psychedelics, such as LSD and psilocybin, bind to the receptor of BDNF, which is called, I don't remember its name, something about tyrosine, but anyway, it's TRKB. And it's the receptor for BDNF. And the effects of psychedelics on neurotropic signaling, plasticity, and antidepressant-like behavior in mice depend on TRKB binding, tyrosine kinase. And actually, they found out that LSD and psilocybin, compared to other antidepressants, because there's also other antidepressants that bind to this receptor, it has like 1,000-fold higher affinity for LSD and psilocybin than other antidepressants. So that's why we have this huge capacity on psychedelics related to the neurogenesis. So this was actually very beautiful, and I really recommend you to take a look on this paper. So here is one question, like, how is this interaction going? Like, how the microbiota will be interacting with psychedelics, or how will psychedelics will be regulating the gut microbiota, which in turn will develop neurogenesis. This is yet unknown, it's unclear. And as I know that also many scientists are here, I would like to plant that seed to this field. Let's explore another perspective coming also from really great researchers on the field of psychedelics. They are also pioneers on the aspect of gut microbiota-Gut-Brain axis. And here we're basically gonna explore the interactions between the psychedelic experience, the microbiota, and our body. So first of all, we know that we have different phases when the psychedelic experience starts, right? Since the moment that we take the decision that we are going to do a psychedelic experience, we need to know that it already starts there, right? So we must take time to prepare ourselves to read about the molecule that we're going to use, or in this case, for therapeutic purposes, also people need to be informed that they're going to get through certain kind of therapy. However, there must be the case that when we know that we're going to do a psychedelic experience, our body is not in the best shape. And actually, these scientists explore that it would be a good idea to actually have certain considerations to shift this pathologic or inflammatory condition into a condition that actually benefit the psychedelic experience. Here I just put that, yeah, if we have inflammation or disease, we will have an imbalanced gut microbiota. But if we know and we are prepared, we can actually do certain things. I'm going to talk about that later on to shape that gut microbiota. And there's actually one of those aspects is the diet, right? There's also research that point out already that they compared two diets: the fast food diet and the Mediterranean diet. And they saw that when they changed diets, after 4 days only, they could already see that the microbial metabolites related to neurogenesis, because there are other bacterial metabolites related to neurogenesis, are already being shifted. So for the people having the fast food diet, these microbial metabolites are being diminished, while in the Mediterranean diet, which is basically based on plants and some fish, these neurogenetic, microbial, and metabolites are being increased. So already from this moment, neurogenesis is happening. Then we have the stage in which we are basically gonna take the psychedelic agent, and we will gonna get some altered brain activity in that moment. All our gut-brain axis is going to be stimulated, as we talked about it before. Our cells, our tissue is going to be stimulated. There's going to be alterations in our microbiome and gut-brain axis. And of course, here we can also have the microbial metabolism related to tryptamines, such as tryptophan, kynurenine, and catecholamines. As I say, catecholamines are involved in the fight-flight responses, and of course, tryptophan is precursor of of serotonin. And also dopamine is really, really important, and is also synthesized in the gut. I think 50% of dopamine is synthesized in our gut, so that's also really, really important. And but it not only stays there. We have a third stage, which is the integration. And that is after we had our psychedelic experience or the psychedelic therapy. And that is not only the day after, but it is sustained through time. There's also evidence pointing out that the neurogenesis actually takes several days. And maybe we can think that this is part of the microbiota contribution in the psychedelic experience. But this is something not new, right? Like this is what now researchers are talking about, also just giving some ideas, because this is not a original paper, it's more like a paper that is just giving some perspective. And we already know from indigenous tribes that, for example, with the use of ayahuasca, you must go to the jungle to do a diet, right? And it starts the moment that you get there, that you find some things with yourself, that you are detoxifying yourself, having certain foods also. You have a really controlled diet, and you get prepared to the day of your psychedelic experience. And this is, and they already know that they do this to have the best outcome on the psychedelic experience, a more profound effect. And of course, afterwards, it's important that you are not back to life immediately, but you also take some time to integrate, to remember all the messages that happen in your psychedelic experience, right? So as again, it's an integrative approach. It's not only taking the psychedelic, but it's taking into account your physical body, but also what it's in your mind to reduce your stress levels. And then you will see an outcome of healing, right? Then one also, well, part of the ayahuasca experience and some other psychedelics, we also have this vomiting or purge, and great deal of neurotransmitters such as acetylcholine, serotonin, and catecholamines are involved in this process. So perhaps neurotransmitters are now making the stimulation on gut microbes there, because of course vomiting will for sure impact your gut physiology and your gut microbes. So to wrap up and summarize this part, so if we have a balanced microbiota, which will come mostly when you have a diet based on fibers, you are active, you do exercise, you have good stress management, meditation, or just like winding out after working with friends, you know, any activity that will help you to chill. It's important. Of course, good sleep. Then you will have vitality, you will have energy. And of course, since this moment, all your neurogenesis process is going to be on. Whereas if you have an altered microbiota based on high fat or high sugar diets, if you don't move, we are all the time, if we actually work all the time in front of the computer, we don't take breaks actually to move, then that can cause, you know, mental conditions, but also that can be translated into immune metabolic diseases or combinations of them, and translated in low energy levels, low quality of life, and aging. And the question is if psychedelics can help us to do this shift right? This we don't know yet for sure, but I mean, there's a great deal of evidence that that can be the case. So also while doing all of these studies among my life, I've seen that we have like this constant war against bacteria and drugs. We have done a lot of antibiotics, antiseptics, pesticides, and of course, we need them, right? If we're sick, I'm so grateful we have all of this. But it's also true that we have been abusing out of it. And not only on our body, but also in the earth, right? Like there's pesticides that are making the soils unfertile and sterile, that the microbes cannot grow in the earth. And actually what wood quality soil depends on the microbes that are there to metabolize all these nutrients. And it's really, really important to be aware of this. And for that reason, now there's these multi-drug resistant strains, in which if some people have these multi-drug resistant strains, they will definitely gonna have a hard time living, because it's very hard to kill these multi-drug resistant strains. And that's why they're developing microbial therapies, such as fecal microbial transplant. But this is just ongoing, and we have to be mindful about it. And of course, there's a huge war against also drugs and psychedelics, which we know that it's just not helping us, that it has bringing a lot of violence, a lot of intolerance. So perhaps we have misdefined from the beginning who our true enemy is. So there's a lot of gaps in the fields. So the Microbiota-Gut-Brain psychedelic, we don't know which signaling pathways are involved yet. And that brings me to ask me so many questions, and also I would like to hear your questions. I would like to hear your input. But one of these questions is like, are the anti-inflammatory properties of psychedelics linked to the immune modulatory role of the gut microbiota? And of course, we always have this, like, what happened first, like if it's the effect of the gut microbiota on the psychedelics or the psychedelics on the gut microbiota, we don't know. And to what extent we can shape the psychedelic experience by shifting the habits that govern our gut microbes. And to what extent the psychedelic experience shapes our microbiota and thus our metabolism. And we don't know what's the microbial metabolism of psychedelics. As I said before, when we take psychedelics, are we taking the psychedelics there? Like, are we having a certain metabolite that is causing maybe a bad trip? Maybe, I don't know, but it will be interesting to take a look. And of course, mostly all of this, is this for therapeutic relevance? And I would say yes. And also, I put it here, I really didn't talk about it, but I put here a reference about my last point, which is the endogenous DMT. There is actually, Dean and collaborators talk about this topic, and they basically track the concentrations of DMT in the body, but of course, DMT is really hard to trace. However, they found out that the two enzymes dedicated to create DMT are also in the gut. And they also found out that actually DMT, it is not in small quantities. Actually, it is in the same comparable levels as serotonin. So perhaps DMT has a huge endogenous role in our bodies, and we don't know yet. But perhaps it's really linked to microbes. Like, can we actually, can microbes degrade DMT, for instance, or are any concentrations of DMT shape any kind of microbiota composition? Yes, and So I have some conclusions, but of course, as I said before, I would like to enrich these conclusions with all of you, because of course this is my perspective, but I know that there are many people here with different backgrounds, and I would love to hear what you think about all of this. We know now, we are aware that the Microbiota-Gut-Brain axis is really essential. It has a really essential role for the therapeutical properties on psychedelics. It is important to consider these 3 stages on the psychedelic experience, because I think it's very helpful to be aware that once our body is in good shape, we will be more able to take that therapeutic properties in a better way, or it can actually potentiate these outcomes. We still need to explore the microbial metabolism on psychedelics due to this microbial conversion of pharmaceuticals. And of course, we have to take into consideration that all of this is a personalized therapy, right? Like, as each individual is different, we cannot assume that we are all the same. And this field is also increasing even more, personalized medicine or precision medicine. That's also important, and it's really beautiful because actually they're looking for multidisciplinary perspective. And I think that that's the approach that we should always have. I would like to give a conclusion, the philosophical one. I wrote something. I wrote, like, the great paradox in science is to see things separately. For instance, we are specialists in few topics because we find a lot of challenge to be multidisciplinary, or we don't know how to do it. And in the real life, it is the same thing. Sometimes we see ourselves apart from nature, when the same natural laws apply to all beings on the earth. The Mayan elders say that to understand well about our own existence, we have to know what is going inside of our body, and they call it the microcosmos. Also, to know what's going on in our environment or in our world, which is our cosmos, and what is going on beyond our world, and that called the macrocosmos. And this is the cover of my thesis, my PhD thesis. And here I just wanted to translate the symbiotic relationship that we have with microbes among all this universe, or all this cosmos of molecules, that even though they're so tiny, it's a whole world, and I can even feel that they are bigger than all. They're so powerful. Here I put a hidden message, which is the DMT molecule. And here are the microbial metabolites. Perhaps the shift is generated from the most microscopic of our being, in this order, dynamic and complex way. And from there, from this tiniest part of us, the changes are being generated in our cells, in our tissue, then in our organs, and then in our whole body. And then it will impact our mind, our emotions, how we behave. And we cannot even think further that it will even touch our spirit. And then the psychedelic experience tries to generate balance in ourselves. It tries to heal ourselves. And when we are in balance with ourselves, it won't only stay with us, but it will keep expanding, touching other human beings. Then it will also impact our surroundings. And I just want to say that what microbes have taught me is to live in community, because we thrive in diverse communities, because we can benefit from the characteristic of each person, each member of the community is unique. And what I have, others don't have, and we can benefit on each other, giving each other this symbiotic way, because in the end, what we want is to preserve the life of others but also to keep safe in the environment that we live and take care of the environment that we live in. So after this, I would like to finish my presentation, but not after saying like, thank you so much for listening to my talk. I skipped some things because I saw the time is a little bit tight just for the presentation, but we still have time for the discussion. I would like to give my deeply thanks to Unimind Maastricht again, to Unimind Groningen, and of course to the Psychedelic Society Groningen, to the Summer School on Psychedelics as well. And I'm very, very, very curious to hear about what you think about this topic.